Bismillahirrahmanirrahim. Assalamualaikum. Good morning and welcome to the fifth Colloquium of Academic and Research, Quarter 2025. The session will begin shortly at 9 a.m. sharp. Kindly ensure your microphones are muted and your cameras are on for the group photo session later. Enjoy the background music while we wait. Bismillahirrahmanirrahim. Assalamualaikum and salam sejahtera. Yang berusaha Associate Professor Dr. Aizi Nurmazila binti Ramli, Dean Faculty of Industrial Science and Technology, Technologist Dr. Azizan bin Ramli, Deputy Dean Research and Postgraduate Studies, Technologist Dr. Hanidah binti Abdul Aziz, Deputy Dean Academic and International Affairs, Technologist Dr. Muhammad Hafiz bin Mazwe, Deputy Dean Student and Alumni Affairs, Associate Professor Dr. Tan Suat Hian, Director of COA 2025. Our respected colleague and co-host from Central Luzon State University CLSU, Professor Dr. Ravelina R. Velasco, Vice President for Academic Affairs CLSU, and Associate Professor Dr. Hazel Monica Matias Peralta, Acting Dean College of Fisheries CLSU. Distinguished speakers from CLSU, Associate Professor Dr. Franz Mariel Garcia, Director, Crop and Resources Research and Development Center CRRDC, Associate Professor Dr. Joel Jobinel, Department Head, Food Science and Technology, Professor Dr. Rich Milton Dule, Head, Center for Tropical Mushroom Research and Development CTMRD. And from UNPSA, Associate Professor Dr. Nina Suhaiti binti Azmi, Dr. Muhammad Fazli bin Farida Asras, and Head of Industrial Biotechnology Program, Dr. Nazira binti Mahmud. All respected participants, committee members, faculty members, students, and fellow audience. Ladies and gentlemen, welcome to the fifth Colloquium of Academic and Research, COA 2025. I am Dr. Aisha binti Muhammad Marsin, your moderator for today's event, bringing the theme Sustainable Biotechnology for Food Security. The COA 2025 event aims to serve as an initial collaboration platform between Faculty of Industrial Science and Technology FIS, University Malaysia Pahang Al Sultan Abdullah, UMPSA, and Central Luzon State University, CLSU. Two key areas will be explored through the COA platform today, which are academic discussions and research within the shared expertise domains of both institutions. To begin, I invite the Dean of FIS, UMPSA, to deliver the opening speech and officiate COA 2025. Please welcome Associate Professor Dr. Aizi Nurmazila binti Ramli.
Thank you, Dr. Aisha. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat sejahtera and a very good morning. Yang berusaha TS Dr. Azizan bin Ramli, Deputy Dean Research and Postgraduate Studies. Technologist Dr. Hanida binti Abdul Aziz, Deputy Dean Academic and International Affairs. Technologist Dr. Muhammad Hafiz bin Mazwe, Deputy Dean Students and Alumni Affairs. Associate Professor Dr. Tan Swat Hian, Director of COA 2025. Our respected colleague and co-host from Central Luzon State University, CLSU, Professor Dr. Ravalina R. Relasco, Vice President for Academic Affairs, CLSU, and Associate Professor Dr. Hazel Monica Matias Peralta, Acting Dean, College of Fisheries, distinguished speakers, all respected participants, committee members, faculty members, and students. On behalf of the Faculty of Industrial Sciences and Technology, University Malaysia Pahang, Sultan Abdullah, it gives me great pleasure to welcome all of you to the 5th Colloquium of Academic and Research COA 2025. This year, we are deeply honored to co-organize this colloquium with our esteemed partner, Central Luzon State University, CLSU, Philippines. This collaboration continues the legacy of the COA series, which began in 2021 and has seen productive partnership with institutions across Brazil, Indonesia, and the Philippines. This year's theme, Sustainable Biotechnology for Food Security, is timely and significant, reflecting our shared commitment to address pressing global challenges through research excellence, academic synergy, and cross-border collaboration. The goal of this colloquium is to foster bilateral knowledge sharing in three major research fields: food science, technology, and agriculture, bioprocess and enzyme technology, and natural product and biomedical technology, as well as academic program implementation with a dedicated focus on biotechnology and agriculture. It is also in line with the strategic thrust of UNPSA 2021 to 2025, specifically Thrust 1A, which is producing holistic graduates, Thrust 3A, generating new knowledge, and Thrust 6A, internalizing student experience. We believe that through this initiative, we are laying the groundwork for broader long-term collaboration such as international student exchange program, global industrial training, global classroom initiatives, and joint international research projects. This inaugural collaboration between Faculty of Industrial Sciences and Technology UNPSA and CLSU also demonstrates how aligned academic structure and expertise, particularly in biotechnology, can open doors to future joint academic offering and research diversification. To our distinguished speakers, your presence today enriched this platform, and we are grateful for your contributions in advancing our shared aspirations. To our organizing committee, your dedication and meticulous planning have made this colloquium possible. To all participants, I encourage you to actively engage, share your ideas, and build new networks. With this, it is my honor to officially officiate the 5th Colloquium of Academic and Research, COA 2025. May this event be fruitful, enlightening, and a catalyst for enduring academic collaboration between UMPSA and CLSU. Thank you. And with that, wabillahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you, Dean of FSDI, Faculty of Industrial Science and Technology, UNPSA. Now, we would like to invite our esteemed guest from CLSU, Professor Dr. Ravelina R. Velasco, Vice President for Academic Affairs. The floor is yours.
Good morning, everyone. Can you hear me?
Yes, loud and clear.
All right. Good morning to everyone. I am very excited to be part of this fifth colloquial event for academic and research. And to our esteemed colleagues, respected guests, dedicated faculty members, passionate researchers, and brilliant students of University Malaysia Pahang, Al Sultan Abdullah, and Central Luzon State University. Welcome to the fifth Colloquium of Academic and Research. It is truly an honor to virtually join you today before a community that shares a common passion, the pursuit of knowledge, the commitment to discovery, and relentless drive to contribute meaningfully to our society through academic and research excellence. This colloquium is more than just a gathering of minds. Do you agree with me? Okay. It is a celebration of intellectual curiosity, a platform to showcase innovative work and opportunity to forge new collaborations that cross boundaries, disciplinary, institutional, and geographical. As we open this colloquium, let us be reminded that the work we do in classrooms, in laboratories, in the field matters. Research is not just about answers, it is about asking better questions. It is about seeking truth, improving lives, and shaping the future. We are deeply honored to have you with our distinguished speakers from both university, from CLSU and UMPSA. Is that right, UMPSA? That's the short name for your university. Have I pronounced it well? To let this event inspire us to think deeper, aim higher, and work together toward impactful outcomes. May we leave this colloquium not only with new knowledge, but also with renewed purpose. Thank you, and once again, welcome to the fifth colloquium of academic and research. Good morning, everyone.
All right, thank you, Professor. Now, may I invite everyone to participate in our photo session. Please turn on your cameras for a group photo within these 10 seconds. We will have a simple 3 snapshots. Okay, so technical is ready. Okay, please smile. Okay, screenshot in 1, 2, 3. Another one. 1, 2, 3. And last one for this upcoming session. Please thumbs up, please. Okay, 1, 2, 3. Okay, now let's enjoy a short montage from UMPSA and CLSU.
What is UMPSA? We are education. In its essence, UMPSA builds the ongoing future generation. It provides the best quality of studying for its students to experience and gain new knowledge. We are passionate, pushing ourselves to become someone who inspires innovation and dedication to continue the legacy. We are technologies, enabling all the world's thinkers to enlighten and shape the world in multiple possibilities. We are TVET, producing quality skilled human resources with their requisite values and beliefs. We are research, exploring new methods and knowledge to develop amazing wonders that will assist our society. We are entrepreneurs. Modern society demands innovative and heart-driven people. UMPSA teaches its students the foundation of what it takes to become one. We are an opportunity, a place for students to create and present their inventions and ideas to the world. We are global, forging a path on an international level through partnerships and understanding. We are a community. Students from across the world can study and interact with ease and share their cultures and heritage. We are service, providing the best facilities for our students to flourish and a chance to give back to society. We are sustainable. For years, the Green Campus has been used to incorporate environmental management and safeguards into tertiary institutions, making them a more livable and sustainable place to study. A place to relax your mind while studying the course of your desire. In UMPSA, anything is possible as our students continue to grow in the present and the future. We are UMPSA.
Babalik pa rin sa yakap mo hanggang sa uling palagi. Pipiliin kong maging sa'yo. Ulit-ulitin man, nais kong malaman mo, iyo ko.
Tanging ikaw ang sigaw ng puso araw-araw. Aminin ko na sa'yo, ngayon lang nagkaganto. Ako'y babalik, babalik, babalik pa rin sa'yo. Oh, 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 oh.
Ladies and gentlemen, a gentle reminder to all presenters today. Each speaker is allocated a maximum of 15 minutes for their presentation to ensure the event proceeds according to schedule. To assist with timekeeping, a bell will be rung once when there are 2 minutes remaining, and again when your 15 minutes have ended. We kindly ask all presenters to conclude their speech promptly to respect the time limits and allow the program to run smoothly. We appreciate your understanding and cooperation. For the audience, you are allowed to make smiley, applause, or thumbs up reaction after every session with supporting comments to show your interest in the topic that has been covered. Now first, let's begin our first presentation. Please welcome Associate Professor Dr. Aizi Nurmazila binti Ramli, Dean, Faculty of Industrial Science and Technology, UMPSA, with the topic Mushrooms for a Better Future, Bridging Food Security and Therapeutic Innovation. Please welcome.
Thank you, Dr. Aisha. So, may I know who will share the slide? Me or the secretariat? The committee? Who will share? Me? Apa share? Okay. I will share the slide or the committee will share?
Dr. Aizi can manage the slide.
Okay, thank you. Okay. Okay, can you see this? Can?
Still not appear, Doctor.
Maybe Secretariat can share the slide for me. Is it okay?
Okay, I will help.
Thank you. Okay, Bismillahirrahmanirrahim. Assalamualaikum. And very good morning to all of the participants for today's COA 2025. So for today, I will present about the mushroom for a better future, bridging the food security and therapeutic innovations. While the committee trying to share my slide, I will continue with the presentation. So I would like to highlight a little bit about the global challenge nowadays, which is talking about the food insecurity and rising chronic diseases. So many people in the world don't have enough healthy food, which is considered as food insecurity. At the same time, more people are getting sick with diseases like diabetics, cancer, and also obesity. So for that reason, we need better food solution to solve both problems. So we need for sustainable, multifunctional biological resources that we call it as eco-friendly, sustainable, and can do many jobs, which is considered as multifunctional. So biological resources is the living things like plants, fungi, including mushroom. So we need to use these natural sources wisely to fix food and health issue. So mushroom actually is underutilized superorganism. So mushroom are very powerful and useful, but people are not using them enough. They can give us food, medicine, and even can help the environment. Superorganism means that this mushroom can do so many things at the same at all at once. So as you can see in this table, okay, so I summarize about the function of the mushroom as a because mushroom contain high in protein, including fiber, vitamins, so it is considered healthy and sustainable for food source. Other than that, mushroom also is considered as medicine because it contains compounds that fight cancer, boost immunity, and protect the brains. So mushroom also can break down agricultural waste, turning it into food or compost, and improve soil health, absorb pollution, and support forest ecosystem. Other than that, it is also used in pharmaceuticals for the biodegradable materials and even packaging. So in the next slide, I show you some infographic about the mushroom as superorganism, including for the food, medicine, soil and environment, and also for the waste recycling. Now we move to the next slide. Okay, mushroom as sustainable food source. So for your information, mushrooms grow very quickly. Some varieties mature in just a few days to weeks. This is very amazing basically. They don't need much space and they can grow even vertically or in stacked trays. Mushroom also require very little water compared to crops like rice or livestock. So it is also high in protein, fiber, vitamins, and minerals. Mushroom considered or categorized as nutrient-dense plant. They provide plant-based protein, dietary fiber, B vitamin, and minerals like potassium and selenium. So for that reason, mushroom is called a functional food. What is functional food? Meaning that mushroom can offer both nutrient and healthy benefits. So it also can fight malnutrients because mushroom can be a healthy alternative to meat. Okay, if we compare mushroom to the comparison with we compare it with traditional agriculture, Traditional crops like wheat or corn use a lot of land, water, and also chemicals, including pesticide and fertilizers, but not for mushroom. We can grow mushroom without soil, okay, in limited sunlight or chemicals, and often use agriculture waste as a substrate. So for the urban farming and circular economic potential, mushroom can be grown in cities. So because of they thrive on organic waste like sodas, coffee grounds, or straw. So this fit perfectly into the circular economy models, which is reuse, recycle, and also regenerate. So let's move into the next slide. So mushroom, as you can see here, I try to show you the most common edible mushroom in the market, which is Pleurotus extraudatus or oyster mushroom. This is very, very common actually because this mushroom grow very fast, easy to cultivate, and especially on they also can grow on agriculture waste. And this mushroom contain a lot of protein, fiber, vitamin B, and antioxidant. And the second one is Lentinula edodes or shiitake, known because of its deep umami flavor and immune boosting compound like lentinan. And it is also used in traditional medicine and modern supplements. The third one is Agaricus bisporus or button mushroom. It is also most widely consumed mushroom globally, include the white buttons, cremini, and portobello, which is versatile in salad, soup, and also for the grilling. Now we move to the next one, next slide, I mean. So I made some comparison, okay, in terms of the nutrient for the common edible mushroom, where all three are low calories, nutrient dense food, and ideal for healthy diet. Oyster mushroom are great for the protein and fiber, while shiitake mushroom contains special immune boosting compounds like lentinan, and button mushroom are widely accepted and rich in selenium. For example, it is a key for the antioxidant. Okay, now we move to the next slide. Okay, for the mushroom cultivation for food security. So mushroom actually can be localized production that can reduce the food mass. Mushroom can be grown locally even in small spaces. So for that reason, it can reduce the food mass. Food mass means that the distance for the food to travel from farm to plate. So by reducing food mass, we also can cut the fuel use and also the fuel emission. And it is also income source for the rural communities because for us to grow the mushroom, it is very low cost and low tech. So it is ideal for rural livelihood. And they also use agri waste for the cultivation, which is they use rice husk, sawdust, banana leaf, and even the coffee grounds, which can turn trash into treasure that can reduce waste while producing food. And it is also the great example of circular economy model. And then other than that, I put it in this slide, it use microforestry and agroecological integration, where mushroom cultivation will support the food security through local, sustainable production, income generation, and smart use of waste, all while improving the ecological health. Okay, now we move to the next slide, which is I would like to explain a little bit about the mushroom in therapeutic innovation. So overview of medicinal mushroom. Certain mushroom has long been used in traditional medicine, especially in Asia. So modern science is now validating these users through clinical and laboratory studies. For example, include Linosus rhinoceros, which is also known as tiger meat mushroom, Gardnerella lucidum, okay, or reishi mushroom, Hericum erinaceus or lion mane, and also Cordyceps sinensis. And some of the bioactive compound in mushroom, including beta-glucan for the immunity, terpenoid, which is good for the anti-inflammatory and anti-cancer, and also Hericanones, which is for the neuroprotective. Okay. let's move into the next slide, which is the biotechnological advancement in mushroom science. So I conclude here there are some advancement in mushroom technology or mushroom studies, which is fermentation technologies to control the cultivation of the mycelium of mushroom-derived pharmaceutical, which include the extraction and refinement of specific bioactive compound from mushroom for the drug development, nanoparticles delivery using fungal polysaccharide that use mushroom bioactive compounds from mushroom, for example, beta-glucans, to form or coat nanoparticles that deliver drugs more effectively, and synthetic biology application that involve mutation and genetic engineering to produce the genes, specific genes from mushroom into other microbes like yeast or bacteria. Okay, next we move into the next slide. Why mushrooms matter for our future? Mushroom as cornerstone of sustainable development. Mushroom also can bridging nutrition and health through integrative science. And the next one is the innovation opportunities in food, pharma, and also environment. Okay, now I would like to share a little bit with the audience today some research and innovation that happened in UMPSA, specifically in Faculty of Industrial Sciences and Technology regarding the mushroom. So this is the first one is the cendawan nanas, okay, or pineapple mushroom. This is the pineapple medium oyster mushroom that used the substrate innovation, and this project starting around 2014 to repurpose pineapple agriculture waste, that we call it as pine peat, as a cultivation substrate for oyster mushroom. This substrate replaced traditional rubber wood sodas and achieved equivalent yield and quality. So as you can see in this slide, I try to show you some of the species of mushroom that successfully cultivated using pine peat or using pineapple medium. For example, the first figure is the wood ear mushroom or black fungus. And the third figure is the variety of oyster mushroom, including yellow oyster, okay, and pink oyster, and also the grey oyster mushroom. And then the next one is the Ganoderma, and the last one is the split gill mushroom. So this is the example of the species that have been successfully cultivated using pineapple waste as a medium. Next, we move to the next slide, which is the cendawan nanas or pineapple mushroom, that we commercialize this mushroom and we transfer this technology to the community so that the community can try to market this mushroom, and then that can cover SDG, Sustainable Development Goal number 1, and SDG number 2, which is no poverty and zero hunger. Let's move into the next slide. The second innovation is the cultivation of Lichnosus rhinoceros, or also known as tiger meat mushroom. So in the next slide, I show you, okay, next slide, I show you the rhinoceros sample collection and cultivation in laboratory, where figure A is the control, is the collected Lichnosus rhinoceros from Lipis Pahang, which is one of the state in Malaysia. And then the sclerotium of Lichnosus rhinoceros in figure B, and figure C is where we do the tissue culture cultivation on agar medium. Let's move to the next slide. So this is the outcome of the cultivated tiger meat mushroom in soil. As you can see, after we did the tissue culture, we transfer the mycelium into the mushroom bag, and then we put it in the soil. And this is the resulted sclerotium of the tiger meat mushroom. The duration of the cultivation in soil is between 3 to 6 months only, and the size and the quality of the sclerotium is comparable to the wild type, the one that we harvest from the deep forest. Okay. Next, we move to the third innovation, which is the cultivation of Cordyceps sinensis. So Cordyceps sinensis is a rare and valuable medicinal fungus traditionally used in traditional Chinese medicine. So this fungus or this mushroom can boost energy and stamina. enhance respiratory function, and can strengthen immunity. So this fungus also, the Cordyceps sinensis also is found can uh, have the properties of anti-inflammatory and also antioxidant. So due to its scarcity and high demand, cultivation in laboratory setting using modern biotechnology is increasingly practiced as a sustainable alternative. So next slide, I show you, this is the findings from the laboratory cultivation of Cordyceps sinensis, where we found that the mycelium of the cultivated Cordyceps sinensis is comparable to the wild type. The wild type is the one that we harvest in the Tibet, Himalaya. Okay, so that's the third, all the studies that we have done actually in Faculty of Industrial Sciences and Technology, UMPSA, that I would like to share with the audience today. And for the conclusion, the dual role of mushroom can serve as a both nutrient rich food and evidence-based medicine that can bridge the gap between nutrition and therapeutic health. The need for holistic solution, rising health and environmental challenges require integrated approach. Mushrooms support sustainable food system and preventive healthcare. And last one is the call to action for the advanced research into functional and medicinal fungi. We also hope that some policies can be made that recognize fungi in health and sustainable frameworks, and also can engage communities to raise awareness and also to promote adoption. So with that, thank you very much. I hope you enjoy my presentation. Thank you. Any question?
Okay,
Sorry
thank you,
for the
Dr.
hiccup. Okay.
Mazila.
Thank you.
Okay, now we open the floors for question. Please raise your hand or type in the chat within this minute in all platform providers. In Google Meet, we have YouTube and also we have Facebook Live.
I have this question.
Yeah, please.
Is there a daily limit of mushroom intake in human diet?
Okay. Yeah. Limit for daily intake. Everything have limit actually. So please do not overconsume, because some of them, maybe some of our body cannot adapt to the some of the certain bioactive in the mushroom, especially for the edible mushroom. And for the medicinal mushroom, for sure there are some limit for us to consume. So you need to ask for the expert, okay, or dietitian on how much you need to consume, okay, for your to get the benefit of the mushroom, for sure. Okay, I hope I answer your question.
Okay, thank you.
Thank you.
Okay, is there any other question from the floor?
Okay, thank you.
Okay, thank you for the question and thank you, Dr. IZ, Dr. Mazila. Now, next, we welcome Associate Professor Dr. Franz Mariel Garcia, Director, Crop and Research and Development Center, CRRDC from CLSU, to present Sustainable Agricultural Technologies and Community-Based Solutions in Rice Farming. Please welcome.
Hello, good morning. Can you hear me? Am
Yes,
I
loud and clear.
clear?
Yes.
All right. Thank you very much for this invitation, and I appreciate the first presenter on mushroom. Later, I think my colleagues will also be sharing their mushroom research experience. So, let me try to share my slide. Okay. If the technical can assist me, that would be great. All right. Thank you very much. So that we have the same time zone. So for this morning, I'll be presenting a bit less technical on rice farming related to biotechnology and other technologies that we apply in order to achieve a sustainable agriculture. So hence the title Sustainable Agriculture.
sustainable agricultural technologies and community-based solutions in rice farming. So I will be presenting different scenarios in the Philippines and how our university is trying to contribute to attain sustainable agriculture. All right. So just a backgrounder, how much of the Philippines produce total rice production? So, if you are aware of our university, we are located somewhere here in Region 3. So, you'll see most part of the Philippines are rice producing areas. So, all those green parts and the darker ones have larger areas of rice production. So globally, putting it in global perspective, you'll see here a trajectory of 2020 rice production until 2030. So a 10-year rice production trajectory along with the demand. The green line shows the global rice production. And the orange line shows the global rice demand. So the, the space in between those two lines shows the surplus or the deficit. From 2020 to about this year, we still have a surplus of rice. But by the time of 2027, going upwards to 2030, we will be experiencing a deficit in rice supply. So hence, it is really important to continue improving rice varieties to address different challenges. So in the next slide, I enumerated here different challenges in rice farmings. First, in terms of environmental, we have water irrigation. So even if you see in the Philippine map, we have various rice production areas, but we still have this problem of water source. So it can be the national irrigation, rain-fed areas, or areas experiencing problems in abiotic stress. Also, we have soil health problem. So the challenge in rice production is the extensive use of fertilizers. So our soil receives degradation over time. And then, of course, we know that rice production has a challenge on carbon dioxide emissions. The next challenge on productivity is the occurrence of pests and diseases. So we have various pests and diseases that occur in dry season and different set that occurs during the wet season of rice production. Now going to the socio-economic challenges, in the Philippines, farmers usually own less than 1 hectare, and the big farmers are those with around 2 to 3 hectares of land for rice production. So it is still considered small area. And we also have a problem when it comes to market. Some of the rice that we produce have low quality. So even if we have high production, if the quality, rice quality is not good, the market cannot buy those kind of rice. And another problem posed by these smallholder farmers is the high cost of production. So still there is a problem how to minimize the cost of production for every kilo of rice in the Philippines. Now, I will be transitioning immediately to biotechnology in rice farming. So one of the challenges in the Philippines is vitamin A deficiency among children. So if you are aware, uh, golden rice developed in another country way back in Germany. It was deployed in developing countries like ours. And because of the issue in vitamin A deficiency. However, since golden rice is known as a genetically modified crop, it has to be regulated. So for a very long time, it is already present in the Philippines, but it remains in the laboratory and yield testing in the confinement of rice research institutes. However, um, to bring this, to bring the researchers of golden rice in the Philippines, to bring this in the community, they really pushed for the um, upholding the regulations of genetically modified organisms. So we have in the next slide, all right. In July 2021, researchers are very happy to have the golden rice approved for commercial planting in the Philippines. However, very short period from 2021 to 2024, the Philippine court ruling halted the propagation, the commercial planting of golden rice because of a lawsuit brought by different um, groups who are working against GMO. So not only golden rice, but also the BT eggplant or the BT talong was affected by the Philippine court ruling. So in another aspect of technology from golden rice, in order to address sustainable agriculture in rice farming, we have this alternate wetting and drying. I'm pretty sure this is also practiced in Malaysia. So you just simply insert a round pipe with perforated holes where you don't need to flood your rice farm the whole production period, but you just have to maintain a certain level of water depth in order to maximize the growth of your rice while minimizing use of water. So this is widely um, spread by different agricultural institutions in the Philippines. And this one is promoted to reduce the carbon dioxide emission. For the next technology, here in our university, we are developing special purpose rice. So we have the special aromatic rice, pigmented rice, and glutinous rice. What you can see in the picture is an aerial view of the different varieties that we are trying to improve. So we're testing them in different yield trials. This one shall address again nutrition and lowering glycemic index among rice consumers. Okay, next slide, please. So part of the breeding that we are doing, the breeding and biotechnology that we are doing, in order to connect immediately the technology that we are developing to the community, we allow farmer input in variety selection. So we allow earlier the farmers to assess the performance of different rice lines. We also deploy seeds for community trials or for location specific recommendations. So one rice variety may not work nationwide. It might only work for certain um, locations. So that is local adaptation strategy. So we can recommend specific variety for a specific area. And this is one of the amazing strategy that Philippines is trying to do when it comes to addressing small holders challenge in rice production. This is called rice production clustering or clustering. So rice production clustering is a strategic approach to modernize the rice industry, boost production, and improve the livelihoods of Filipino rice farmers. So in the picture, you will see the the Department of Agriculture or DA. It launches the first 1,000 hectare rice cluster in southern part of the Philippines, in Oriental Mindoro. So here, the government is trying to provide technologies that will adapt to the whole vast of rice areas to produce the demand in that area or even contribute to the demand in the Philippines. Okay, so part again of rice production clustering, one of the program is the rice business innovation. So it is not enough that we address the production problem, but so we have to link the production to the market. So one of the strategies is through the rice bees or rice business innovation system. So PhilRice, the Philippine Rice Research Institute, links clustered farm groups with private sector. This private sector will buy the produce of the farmers on a pre-agreed price even before the production. The next program is what we call the RCEF seed program, or RCEF stands for the Rice Competitiveness Enhancement Fund. So this program implemented 25 sites across the country that will showcase the benefits of using quality inbred rice seeds to enhance the yield and to reduce the cost of package technologies. And of course, the use of farm mechanization and digitalization. So, emphasizing a farm clustering approach, each site will have 50-hectare rice cluster to bolster and maintain the agri-enterprises developed and engaged by the farmers' cooperatives and associations. So that is one strategy to bring technologies to the community. And in our university, we have this program called Technology Village Development Program. So this program um, highlights the special purpose rice that you saw earlier that we try to breed and improve. So bringing this technology to community, we support the farmers in their production, and we even engage the local government units nearby. So the other picture shows you the different MOA signing with different LGUs in the region. So hopefully we will try to, this will be modeled by other universities as well. So I'm almost at the end of my presentation. In terms of sustainable agriculture, we can really improve food security, have a sustainable resource use, and empowered community. So that is really the model that we need to follow or enhance more. And of course, even if we have this working model, we also have, we still have some challenges that we need to address, especially with biotechnology. We hope that GMO will no longer be too much of a concern, but we will have gene edited crops that will be less regulated or at least be more acceptable to the consumers. So we want all the biotech products to have access and affordability. With that, we also need to address the challenge on infrastructure and training. All right. So for policy support needed, an inclusive biotech policies, invest in rural services, and support innovation systems. So for a takeaway in this presentation, the biotech existence plus bringing it to the community will equate to a resilient um, ecosystem or a resilient community where we can really thrive despite the different challenges that we will be facing. And innovation must be inclusive, plus sustainable food system is not impossible, it is achievable. So thank you very much.
All right, thank you, Dr. Garcia, for the excellent presentation. We will now have a Q&A session. The audience, please proceed with your question. You may introduce a little bit your name and where you come from.
Hello. Hello, I'm Dr. Lee from UMPSA.
Hello.
Thank you very much for a very great presentation. So I'm also working on rice, so mainly on microbes, so relating to the biofertilizer. So I would like to know, you have covered so the use of variety and then how you actually manage the governance, so to improve the rice farming. So how about the waste, the rice straw, any sustainable waste management that actually Philippine practice?
Yes, thank you for raising that. It is I tried to minimize my slides to go with the allowed time for presentation, but I used to have that slide for fertilizer use. So yes, you're right. We're trying to in organic farming. In another aspect of rice production, in organic farming, we try to maximize the use of rice waste, convert them to biofertilizers or other form of organic fertilizers and apply them in organic rice production systems. So that is one way that at least our university is doing when it comes to using rice waste turning into biofertilizers. Thank you for your insight.
Thank you. So do you use microbe to degrade the rice
Oh, yes.
waste?
For personally, I don't have a research on microbes in rice, but I know some of our researchers are using different microbes in rice. Yes.
Thank you. Thank you very much.
Okay, so thank you for the remarkable answer from the panel. Okay, now Dr. Muhammad Fazli bin Farida Asras from UMPSA will share on strategies for optimizing bioprocess efficiency. Please welcome Dr. Fazli.
Right, testing mic. Can you hear me?
Yes, I can hear you.
All
Please
right.
proceed.
And can you see my slide?
Yes.
All right. Thank you so much. Thank you so much, Dr. Aisha, for the invitation for me to deliver at least a 15-minute speech on this platform. First of all, Assalamualaikum warahmatullahi wabarakatuh and good morning. Distinguished speakers from Central Luzon State University, Associate Professor Dr. Franz Maria, Associate Professor Dr. Oil, Professor Dr. Rich Milton, and Associate Professor Dr. Hazel. So I'm so sorry if it is sound freaky. And also my colleagues from UMPSA, Associate Professor Dr. IZ, Associate Professor Dr. Nina, and also Dr. Nazira. Dear Professor, lecturers, students, and also industries and other fellow friends. First of all, I would like to thank to the organizer for the invitation on this special event today. And I also would like to share my experience on the topic entitled, as you can see on the slide, is strategies for optimizing bioprocess efficiency and sustainability through integration of upstream and downstream methods. Okay, dear guests, when we are talking about bioprocess related to upstream and downstream processing, most of us will use this figure. Okay, this first figure, in which you can see that it's showing from the beginning, such as you can see strain development, searching for the raw materials, and up to the end of the product, such as crystallization, packaging, and also delivered to market. And as you can see down here, from the simplest flow diagram above, it can be elevated to the complex flow, especially when we are dealing with a pharmaceutical product or high resolution product, such as in here I showed to you is a monoclonal antibody from the CHO, Chinese hamster ovarian cell. Okay, so maybe from the simplest one to the complex one, from 2 or 3 operation units, so you can have more than 5 units, even though the principles are similar, and sometimes the product are similar. Okay, so as part of that, apart from the laborious work in upstream and downstream processes, as we can foresee, if let's say uh, those money that we have, the normal cost that we need to bear is about 60 to 80% of the total cost for the downstream processing. So just a simple calculation, as you can see here, I posted here. If I have 1 million ringgit Malaysia, or even I have 1 million peso, so as high as 800,000 peso will burn in this process. And the critical question is how we gonna charge for the product and how much we can get it in our pocket. Okay, so is it worth or worthless? We are doing upstream and downstream processing, so that you need to think of before you start manufacturing your product. And this is economic cycle keeps us reminding how sustain our product in the market and also to the consumers. So this is how integration takes place, where we have a check and balance. And one of the simple approach, or you call it process analytic, where we can see the relationship between 3 things. First one is the parameters. Second one is the contaminant that we put in our operation unit. And lastly, the product variant came out from each process. And lastly, we can have the final product. Okay, so let me share our experience on the integration of these processes. Normally, what we have here, a biochemical pathway of our product. Okay, so where it begins and also where you can see your end product. Starting from the simple sugar like glucose, fructose, and others, to the end of your product. Okay, and from this simple sugar, so it goes to other cycles, maybe TCA, G6P, and others. Okay, by looking at this, we can plan the upstream process, starting from the conventional and ultimately to the cutting edge technology. Both routes, conventional or with a cutting edge technology, you will have same end product. The only matter is the time and also cost that you already applied for. Okay, so down here we have a project with the agricultural industry. So you will have abundance of spoiled fruits and other organic matters or agricultural waste. And rather it goes to the dump site and naturally being rotten. We process this gold to something beneficial. Same concept as principle as I mentioned earlier on the upstream and downstream processing. Okay, so last of my 2 slides, I think This is my research interest. Other than that, another major problem in big cities is the dump site or landfill, whether they cater the industrial or domestic waste. I think you have one, the Clark Sanitary Landfill in Capas, Terek, operated by, I think, if I'm not mistaken, is Metro Clark Waste Management Corp. They cater for Clark area and also northern and central Luzon. Okay, so I can see in here, you can see that from waste that you dump to the landfill up to the new landfill, is this waste take years to degrade, and some cannot even degrade, like plastic base, rubber, glasses, and also other material. And the government will end up looking at new place. That's why I put here new landfill, new place to have another artificial hill like this one. Okay, so it is not just 1 acre. I visited this landfill. I think it's more than 20 acres with a contract project with a state government. Okay, so lastly, in here we have a project with Universitas Airlangga to enhance the microbe itself to tackle the organic or the degradable waste to something what we call it organic acid. Okay, so that one will be the high throughput product. From normal route, currently we process this product from the beginning up to the end. It takes about 14 days. Okay, and we are very ambitious and bold to get the product just after 4 or 5 days, including all those hanky-panky processes in upstream and downstream processing. Okay, and as you can see in here, we stress on the upstream part by putting risk on the CRISPR technique and also challenge our team to extract. Currently, we already extracted the organic acid is 0.1 gram per liter out of a 5 gram of waste. That one is our preliminary data. So with that, we are very confident in delivering these high resolution products where all the integration take place by some approaches that you can try to apply on, like quality by the design, real-time monitoring of your process parameter and also your final product, and also some angles on the data analytics. Okay, so that one you can project how much product that you can obtain at the end of your process. With that, thank you so much for your kind attention, and my special thanks to Kyoto University Japan and also Universitas Airlangga Indonesia, and also two industry that I work on, which are Gading Senggara Sinar Berhad and also Alam Flora Environmental Solution, for giving us technical and also scientific support along this way. Thank you.
Okay, thank you, Dr. Fazli. Now the floor is now open for any questions.
I have question. My name is Nurin Sajida. I want to ask, how can process integration reduce waste and energy consumption in bioprocessing?
Okay, thank you, Ms. Nurin. As I told you in my slide, normally when you have a product and you have all those processes in your mind, including your upstream and downstream processing, those will gives you at least 80% of the total cost, 60 to 80%. But I think the major value is 80%. So how you want to integrate is sometimes we try to eliminate same process like the filtration process. You may put three different types of filtration processes. Okay, and also you have, let's say, three different chromatographic technique. So you can eliminate one, just one in a filtration system and one in a chromatographic technique. So you can less or reduce the cost and also time consuming of your product. If I said that the end product will same, okay, whether is a high resolution or low resolution of your product. That's all. Thank you.
Okay, thank you to Nurin for the question. And thank you once again, Dr. Fazli, for the great presentation.
All right. Thank you so much.
Now let's continue with Associate Professor Dr. Joel Jovinel, Department Head, Food Science and Technology from CLSU, who will discuss fermentation and enzymatic dynamics in cocoa. Please welcome.
Good morning. Can you hear me?
Yes, I can hear you.
All right. May I ask the technical staff to present my slides, or I can also present it from my end if you like.
Which one do you prefer? Do you want our technical or you want to present it?
Can we try the technical first?
Okay, technical
And
first.
if it won't work, then I'll present it. And while they are looking for the presentation, I'd like to thank the UMPSA for this invitation. And I'm very happy. And I just want to know, just a show of hands, who loves chocolates? Thumbs up if you love chocolates. Oh, great. A lot of people love chocolates. And yeah, so I'm really excited to share to you my presentation because it's all about cocoa or cocoa and chocolates and the flavor of cocoa as affected by fermentation. Okay, do you have the slides? Great, thank you very much. Oh, I think that's the old presentation. I will present it from my side, I think. Okay. That was the practice slides. Okay.
So our technical team will stop sharing.
Yeah,
Okay.
and I will share. I know that this is live, so please bear with me. All right. Can you see my slides now? I will present it in slideshow.
Okay.
There you
It's
go.
clear.
All right. So the presentation is entitled The Role of Fermentation and Enzyme Dynamics in Developing Fine Flavor Cocoa, or cocoa, as we call it here in the Philippines. Just a few years ago, we saw this small change in a favorite chocolate of ours. This is actually Toblerone, if you recognize. And just a few years ago, they did a small change in their chocolate. And if you notice, the old Toblerone has a smaller distances in between the triangles. And now the new Toblerone, at least in Europe, they marketed this, has a bigger gap. And what is the reason for this? No? This is because of the rising cocoa prices. So they are selling Toblerone now for the same price, but with a lower weight. So the old one has 400 grams, the new one has 300 grams. So what is the reason for this? Because the cocoa industry actually is in crisis. So since maybe 5 years ago, and the most intense year for cocoa was last year, there is a downward trend in the production of cocoa. And this is due to the global warming, aging trees, and diseases and pests in cocoa that have lowered cocoa supply. And therefore, because of the law of supply and demand, it caused a sharp rise in prices of cocoa. But every crisis offers opportunities. No? That is why in this presentation, in about 10 minutes, I would like to share to you just a very brief overview of the cocoa industry, fine flavor cocoa as defined by the ICCO, factors affecting cocoa quality, the steps in processing cocoa and a closer look on fermentation and how it affects flavor of cocoa. And what are the sensory attributes of fine flavor cocoa? And I will give you a very brief, humble summary. If we look at the global supply of cocoa, we see that West African countries dominate the production. No, they are led by Ivory Coast or Cote d'Ivoire, and followed by Ghana. In this in this statistics from Statista, the only Asian countries are Indonesia and Papua New Guinea, if we can consider Papua New Guinea as part of Asia, but it's Oceania. So about 10 years ago, Indonesia was actually third in global production. But because of pests in cocoa, there was a very sharp decline in the production of Indonesia. Now they are only sixth in global production. The cocoa industry distinguishes between two types of cocoa. One is bulk or ordinary cocoa, and the other is fine flavor cocoa. As we said, bulk cocoa is quite ordinary because the flavor is just the basic chocolate taste and it's quite bitter. While fine flavor cocoa has fruity, nutty, sometimes caramelic attributes, and it has more nuanced flavors. So, what is the importance of this? Bulk cocoa actually is priced lower in the market, while fine flavor cocoa can go up to twice or even three times the price of bulk cocoa. So, what is the opportunity in this crisis actually? That we can compete as Asian countries, not in terms of bulk or not in terms of quantity of supply, but by quality of our supply. And I'm happy to share to you, this is the definition of fine flavor cocoa. I will go back to that later. I'm happy to share with you that although international trade is dominated by South American and Central American countries, as we can see, this is statistics from the ICCO, and most fine flavor cocoa is from Central and South America, like Brazil. 100% of their cocoa exports are fine flavor. We have Colombia 80%, Dominican Republic 60%, El Salvador 100%. But the good news is some Asian countries already also produces fine flavor cocoa. There is an ad hoc panel of the ICCO that evaluates if a country has fine flavor cocoa or cocoa. And Indonesia already has 10%. And we have Vietnam at the bottom of the list here. They are exporting actually 80% of fine flavor cocoa, or at least 80% of their exports of cocoa is 80% fine flavor. Well, we can see here Malaysia. No? Malaysia has no percentage yet. It has letter A there, and it signifies that Malaysia is already also exporting fine flavor cocoa, but the ad hoc panel of the ICCO, the International Cocoa Organization, has not yet met to evaluate the percentage of the cocoa. So that's good news for Malaysia. And I also bring good news from the Philippines. Recently, we published together with my colleagues from Ghent University in Belgium a discovery that our own Philippine cocoa beans contains fine flavor, volatile organic compounds. So, and we found this in the south of the Philippines in Davao City, the largest producer of cocoa beans. And we processed this also, and we evaluated the attributes, the sensory attributes of this cocoa, and we had very promising results. I just want to show you the cocoa belt, or the places where cocoa grows or cocoa grows. And as we can see, it's around the equator, no? And as I told you, it's dominated by Ivory Coast, Ghana, and Cameroon. And also in South America, Colombia, Ecuador. But in Asia, it's actually Malaysia, Indonesia, Philippines, and New Guinea that produces cocoa beans. Based on a review article that we did, West African cocoa beans can be are known to have basic chocolate taste, while Central and South American cocoa beans have fruity and fine flavor. Well, the sad thing is, according to published studies from the last 10, 20 years, Asian cocoa beans are most of the time, if not always, classified as acidic, no, vinegar or acidic in its aroma. So that's why our recent publication of the one we published with Oxford Academic is really important, because we saw that we have in Asia, and we know this for sure, that Asian cocoa beans has also fine flavor characteristics, because it is not just a function of the terroir or the place where it is grown. No? It's also the genetic makeup of the cocoa beans. And we have also in Asia, we have the Criollo variety, we have the Trinitario variety, and we have the Forastero variety. Criollo is known as the fine flavor variety, together with Trinitario. Just a short, just a very quick trivia. It is actually, historically, it is actually the Philippines where the first cocoa plant was planted. No? Because cocoa beans actually originated from Central America, from Mexico. And why the Philippines? Well, because as you know, we were colonized by Spain for 300 years, from 1500s. And that's why the the ship that goes to from Spain to Acapulco in Mexico. So they also brought cocoa trees to the Philippines, and it's the Philippines that where the first cocoa plant in Asia was planted, and it spread very quickly also in Indonesia and Malaysia. So at least that's what the historians say. Now let me go to the meat of the presentation. This is really the peak of the presentation. So let's now go to the fermentation process of cocoa and why it's important, and how enzymes are produced during cocoa fermentation. Let me just take a sip of water. All right. So if there's a take-home message that I want to say, it is this. Fermentation is a very crucial step in cocoa post-harvest. If we don't ferment our cocoa, we will not have fine flavor characteristics. Again, I cannot overemphasize the importance of fermentation in chocolate production or cocoa product development process. Let us go to the few steps in the cocoa post-harvest stages. So we have the cocoa tree here, and then normally cocoa pods, that's the fruit of the cocoa, are harvested twice a year. In the Philippines, it's in June and December. I don't know in Malaysia, maybe it's also the same. So the pods are harvested, and then the pods are shelled, so they are opened, they are cracked, and then they are placed in usually baskets. So in Asia, it's in baskets or in wooden storage boxes, and they are fermented for maybe at least 3 days to 5 or 6 days. So they are fermented in wooden boxes, and they are covered with leaves of banana. So in Asia, we have a lot of banana leaves. After fermentation process, the cocoa beans, when they reach a certain moisture content of maybe lower than 10%, they are sun dried. For in tropical countries, because we have a lot of sun, we sun dry our cocoa beans for about a week. Now, the magic happens in fermentation. Why? Because in fermentation, it significantly enhances cocoa flavor by promoting the formation of volatile organic compounds such as esters, aldehydes, and pyrazines, no, through microbial metabolism. And in fermentation, as you can see, it is not just one group of microorganisms that is responsible for fermentation. There is actually a succession of different types of microorganisms. So we begin with yeasts. So in the first day of fermentation, the first 24 hours of fermentation, spontaneous fermentation of Saccharomyces species, Saccharomyces cerevisiae, for example, happen. So the sugar compounds in the pulp of the cocoa uh, is converted, no? Converted into alcohols, different type of alcohols, and then the following day or after maybe 48 hours, we have lactic acid bacteria. No, we have lactic acid bacteria that produces acetic lactic acid, also very important in cocoa flavor. So we have Lactoplantibacillus plantarum, and we have Leuconostoc species. Then, after that, after about 4 days, then we know that we know that alcoholic fermentation is almost always uh, followed by acetic acid fermentation. No? So we have acetic acid bacteria that we have acetic acid bacteria that continues the fermentation using the alcohols into vinegar, into CH3COOH. And the problem is if we continue further with the fermentation, if we let it ferment, no, for a very long time, we will have a problem with spore forming bacteria and fungi. So there is a sweet spot in fermentation of cocoa beans. And it's about 4 or 5 days, depending, of course, upon the volume of the cocoa beans. And temperatures rise up to 50 degrees Celsius, and killing the seed of the cocoa beans. But with this process, polyphenols are reduced, so bitterness is reduced, and also and flavor compounds are produced. Okay, so how do we determine the fine flavor in cocoa? So this is a machine I used during my PhD days. This is an ultra-fast GC nose. So it's an electronic nose. It's actually, the principle is gas chromatography, and this is with FID. So we have the volatile organic compounds such as butanediol, no? And then phenylethylamines that are, and pyrazines actually, methylpyrazines that are responsible for the chocolate and the nutty aroma. And also last year, we did a sensory attributes review of fine flavor cocoa beans. This is with my colleagues from Ghent University again, and one colleague from Indonesia. And what did we see? No, that same cocoa variety, for example, Criollo, Trinitario, and Forastero, share similar flavor notes among varieties from different regions. But there are specific flavors that are unique to certain origins. For example, caramel notes is reported only in Criollo varieties from Mexico. So we don't have evidence yet of the caramel note present in our Asian cocoa beans. Maybe in the future, in our future collaborations, maybe we can find this in our Asian cocoa beans. Nutty notes found in Criollo from Mexico and Peru. And then we have origin-specific flavor attributes, Trinitario varieties from Colombia that produces fruity notes, and Dominican Republic and Indonesia that produces fresh red and yellow fruit notes. So very promising for Asia. And I just want to share very briefly a work of my colleague from an African colleague from Ghent University. Maybe you will be surprised. This is a paper he published about maybe in 2018, and the gist of this paper is this: if we harvest the cocoa pod and we don't open it yet, and we store it for 7 days, we will have a significant increase in amino acids, and therefore it will increase also the volatile organic chemicals responsible for cocoa, for fine flavor cocoa. So just by storing the cocoa beans, we will actually increase fine flavor. So let me end with this take-home message. Fermentation reduces bitterness and astringency by breaking down polyphenols. And we know if we have sugar and then we have high heat and protein, amino acids, we will have Maillard reactions during roasting. And fermentation is brought about not just by one type of microorganism, but a microbial succession of yeast, lactic acid, and acetic acid bacteria producing key volatile organic compounds such as alcohols, esters, and organic acids. And these are the taste precursors that later develop into the chocolate's signature flavor during roasting. And lastly, properly controlled fermentation leads to the development of desirable precursors, essential for the generation of chocolate's characteristic aroma during roasting. And that ends my presentation, and I thank you for your kind attention. Thank you very much.
Okay, thank you, Dr. Jovino, for the interesting cocoa presentation. So, any questions or comments from the floor? We are open.
Yeah, I have a question. Thank you, doctor, for your presentation. I'm Zishan Ahmed from UMPSA. My question is it possible to develop a mixture of microbial starter culture that we can use repeatedly to obtain desired quality of cocoa?
Thank you very much, Zishan, no, for your question. Um, Actually, some studies have done because we know that cocoa fermentation is spontaneous. No, you don't have to put starter cultures. But other researchers already thought about it. That they placed starter cultures with pure starter cultures in cocoa flavor. If I remember it correctly, there are different uh, studies that publish different results. Some studies say that inoculating with pure cultures do not have a significant effect in the flavor of cocoa beans because we have to realize that it's not only although fermentation is important, it's not the only factor affecting flavor. Roasting is also very important. If you roast for a very high temperature for a long time, you will destroy all fine flavor. Other studies say that inoculating with pure cultures produce better quality cocoa because they can manage quality better. So it has been done, and there are still a lot of opportunities for future research for that.
Thank you, doctor, for your answer.
You're welcome.
Okay, thank you for the question. And once again, thank you, Dr. Jovino, for the fine presentation.
Thank you.
We now invite Associate Professor Dr. Nina Sohaiti binti Azmi from UMPSA for a presentation on biotechnological applications of lactic acid bacteria. The floor is yours.
Ya, Assalamualaikum warahmatullahi wabarakatuh. Good morning, selamat pagi, magandang umaga. So I'm Nina Azmi from UMPSA. So today I'm honored to share a Malaysian journey from soil to soul on how community farming and innovation can safeguard our future food. Before I go further, can you see my slides? Is in the maximum of...
Dr. Nina, do you want to share the presentation slides or just the Google Drive? Okay.
Can you see my slides?
Okay.
All right. Okay, so let's begin with basics. Yeah. So what is food security? So food security is about consistent access to nutrition, sufficient and safe food. Yeah, it's not just about having food, but it's about we need to ensure that in terms of availability, access, utilization, and stability over time. But I would like to share with you about Malaysian context. Even in Malaysia, we face food insecurity. Our farmland is shrinking due to urban sprawl, and our farmers are aging, and very few replacement with young farmers. And we depend on the high imports. So these are serious warning signs that we need to bear in mind. And but the crisis goes deeper into our soil. By 2050, up to 95% of land at risk because of the soil degradation, and we lose 24 billion of fertile soil every year. Yeah, so this is very crucial, and this leads to lower yields, poorer crops, and ultimately food shortage. Healthy soil is the foundation of food security, and without it, we cannot grow our future. So, why community farming? Because this is very important. Community farming can strengthen food systems against global disruptions. We can empower community by building skills, create jobs, and fosters collaboration. And it also reduce import dependency, and we can preserve food sovereignty. But the most important thing is how it could focus on how we could promote eco-friendly practice and food literacy, especially in sustainability focus. So in UMPSA or at Biosphere, we not only try to find a problem, but we try to find out a solution in isolating beneficial microbes, and we develop microbial formulation for stable biofertilizer, and we do testing, field trials with various crops, and we also work with farmers. We do implementation, real case scenario. We work with them. We try to find out what is the feedback from farmers. So I also have a company because of university encourage researchers and lecturers to have a business startup. So this is maybe agenda on how we can deliver our products to reach market, to reach community. And Glyco Bio International is my startup company. It is a bio-based company. So we received Super Praju Fund in 2017. One of the innovation, which is probiotic powder, our innovation has been selected among 10 winners, and it has been screened over 800 products in Malaysia. So we also receive local and also international recognition. And the Clever, if you can see there, Clever is our brand. Our tagline, Clever, your clever choice. So our products actually come with two packaging, two forms. We have a biofertilizer plus, it is in powder form, no bad odor and good for soil amendment, and it is suitable for end market. And we also have Bio Booster Plus, it's in a liquid form. This Bio Booster Plus is suitable for commercial scale, for large farm for more yield. So our probiotic formulation has been patented. Because of the scenario that I highlighted just now, because of the issues of soil degradation, the excessive use of chemicals, fertilizer that lead us to come up with these products. So our products have been tested on I believe that more than 117 type of plants. It comes in all-in-one nutrition in one packaging. So it consists of more than 10 billion CFU probiotics in one packaging. It is safe and easy to use and suitable for all plant types. By using this product, we believe that it can reduce chemical reliance up to 30%. It can increase on absorption rate up to 28%. We can decrease the fertilizer cost about 16%. So I show you the results of our innovation. We try to get testimony from our users. Our users among, from Peninsula Malaysia and also Sabah and Sarawak. So they tested the flowering plant, landscape, fruity vegetables, leafy vegetables, and also fruits. So you can see the blooming rate by using our product. Biofertilizer can bloom up to 40%, and the growth rate can increase up to 24%, and fruiting rates up to 18%. The growth rate for leafy vegetables also up to 25%, and for fruiting plants like bananas, melons, up to 15%. So we are not only working in the lab, but we try to reach community. We go out, we to engage. And I also been invited as a speaker in several occasions, such as one of the community talk organized by Bioeconomic Corporation. And I also involved in what we call a road tour, or what we call event organized by Dewan Bandaraya Kuala Lumpur, because this is very important for us from university to educate, to reach people, to reach community. But maybe some of them, they don't aware about our technology. So because university, this is a platform for us to do research, innovation, and how about people out there? So this is the time for us to go there, to go out, and we can share. So if you look at the figure, my picture number 2, this is during my talk at Rimba in town KL. I also managed to give several sessions, talk to single mother, organized by NCWO, and I also appointed as a visiting professor at Prince Songkla Tyler University for 2 years term to give talk on sustainability. So Clever Showcase Farm at UMPSA, we do testing, we do research in the lab, but it's the time for us to show and test on the real plot. That's why at Clever Showcase Farm, UMPSA give us 3 acres land for us to build, for us to test. We call it as a living lab. And we can test with the different formulations of fertilizers, different substrate, different microbes consortium we can do. So this project, this showcase farm, is actually integrates cross faculty, not only from our faculty, but we also work together with faculty from computer science, from electronic, from industrial management. So from there, we can study from scratch until the end output. Yeah. So this one is a community project at Rapta. Rapta is actually Rumah Penyayang Tun Abdul Razak in Pekan Pahang. This is actually the place where poor people, family background, students from poor family background, who lives in a rural area. So this is the place where they stay, they live, they make like their own house. And Rapta is actually in town and easy access to their school. So we try to engage with them. We actually prepare modules. We already delivered modules to them on what is the fundamental of the urban farming, how to grow plants, what is biofertilizer, and all the techniques that we have for urban farming, such as hydroponic, fertigation. And you can see they are very passionate. They are very passionate on doing their tasks. So they try. We actually doing by ourselves. We assemble and disassemble together. DIY. Yeah. We buy pieces by pieces from outside, and we assemble together. So they are very. They were very smart kids actually. You can see the pictures there. How they work with the projects. And in Rapta, we also have fertigation, hydroponic, and also tilapia. Maybe we have pond. They just built pond, and we help them to make sure that the condition of pond is okay. We studied about the fish, and this could give them a good income generation for them to use to manage the raptor itself, and also for them to continue their life and sustain at raptor. Okay, you can see there. So the smiley face, yeah, the smile. So we can see they are very happy working with the farming and our projects. So you can see the spinach, yeah, and also the leafy vegetables, and we engage with them every week. We go to Rapta to see them, to deliver modules, to hands on with them. Yeah, of course, a lot of obstacles actually, because of to do farming is not an easy task. A lot of challenges, depends on the weather, depends on the other issues, so but we manage to do that. So our vision, we start with the soil health, the problem, yeah, the foundation of sustainable food system that come from the soil health. And we try to build resilient communities with knowledge sharing and self-reliance. And we try to bridge science and society and research that serves real needs. And we nourish the soul to connect land, food, and each other. So our journey during exhibitions and collaborations, we also have engagement not only communities but also with government agencies, industries. If you look at the picture number 3 from bottom, is actually our collaboration with Central Sugar Refinery, where we develop the product for them. They have the sugar processing waste, and we try to develop a biofertilizer for them. Actually, we try to formulate based on different conditions, so different scenario and different waste has different formulations. And we also attend several exhibitions such as SIF Korea, ITEX, Bio Tracks, and many more. I said. So we also share our think, our products, our innovation with communities through newspaper. So Pertubuhan Pengguna Islam Malaysia PPIM also try to use our products. They tested on paddy, and they try to grow paddy in pot. They successfully executed the paddy pot by using our clay bar. And second picture is Rimba in Town, where Clever has been one of the most popular booths in Rimba in Town, and also give talk to them about probiotics. Pasar Tani, we always go to Pasar Tani because I think we need to meet people. We need to deliver a message. We need to talk and share about our innovations. And we also involved in Pasaraya Kuala Lumpur, organized by Dewan Bandaraya Kuala Lumpur. So call to action, we start from small, begin with container, garden or community plot. We can learn together, join knowledge or sharing networks. We can adopt sustainable practice by soil-friendly approaches. And we also can connect, reach out to join our initiative. You can also reach out to join our initiative, and you can contact me, Nina, at umpsa.edu.my. So for closing remarks, I would like to say thank you very much to Rumah Penyayang Tun Abdul Razak, to my friends, to my students, to my FIS collaborators, to UMP, to UMPSA, to industries, to all collaborators, because of the effort and passion that they put throughout the projects. And please join us to expand the mission, in future. And my philosophy is return to soil, not just to plant, but to heal. Okay, so let's grow food, hope, and communities together. contact me, Nina at umpsa.edu.my, and you also can reach our Facebook page, Clever. Thank you.
Thank you, Dr. Nina, for the tremendous presentation from soil to soul and sharing her clever innovation. The floor is yours for any upcoming questions from the audience.
Thank you for sharing by Dr. Nina. I have one question. How often do the farmers or the users need to replenish the soil nutrient with probiotic fertilizer?
It depends on the conditions of their space. So, maybe twice a week, twice once a week, so they can make sure that they consistently treat the soil, but it depends on the situation, how big issues of the soil. If let's say the soil is okay, maybe you can just treat it maybe once a month, something like that. It depends, yeah, how crucial it is. Yeah. Thank you. Any question?
Okay. Okay, I think it's okay. Thank you again, Dr. Nina Suhaiti, for your presentation.
Thank you, everyone.
Next, we have Professor Dr. Rich Milton Dule, Head, Center for Tropical Mushroom Research and Development, CTMRD from CLSU, presenting Mycofutures, the next frontier in mushroom biotechnology. Please welcome Professor.
Hello, good morning, everybody. Okay, so let me share my presentation. Okay. Do you see my presentation? Okay.
Okay, we can see your slide.
Okay. Okay. So can you hear me?
Please proceed.
Yes, sir. Okay. So a pleasant day to everybody. So good morning. First of all, I would like to thank the organizers of this international colloquium for giving me this opportunity to share our activities, research activities, as well as our accomplishment at the CLSU Center for Tropical Mushroom Research and Development. Okay, so my talk this morning is entitled Mycofutures, Unlocking the Next Frontier in Mushroom Biotechnology for Health, Industry, and Sustainability. Okay, so what are mushrooms? Mushrooms are Basidiomycetes or Ascomycetes, Hypogeus or Epigeus, macrofungi that comprise a system of branching of mycelia and fruiting bodies which can be seen by our naked eye. And each mushroom has its unique morphological characteristics or morphological structures. Some mushrooms resemble a star, corals, bird's nest, rat's ear, and other several forms. And some mushrooms are considered poisonous, and some mushrooms are considered medicinal and edible. They have been traditionally until recently exploited for their pharmacological and pharmaceutical purposes. And mushrooms, as we all know, are very nutritious and highly medicinal. So they are rich sources of proteins, carbohydrates, dietary fiber, ergosterol, minerals, and vitamins. So apart from their nutritional values, mushrooms are also considered a very good source of biologically active compounds, including the polysaccharides, bioactive proteins, phenolic compounds, carotenoids, tocopherols, ascorbic acids, and terpenoids. So the Philippines is rich in diverse mycological resources. So with our paper, a lot of our paper has been were published in order to, of course, document the different mycological resources in the Philippines. And this most of these mushrooms still remain in the wild, waiting to be discovered and harness their promising potential for pharmaceutical or for other applications. And these mushrooms remain to be pharmacologically unexplored, which warrants special attention. So that is why the Center for Tropical Mushroom Research and Development of Central Luzon State University which is considered as one of the Tuklas Lunas. When you say Tuklas Lunas, that's the drug discovery development center in the Philippines that investigate the Philippine wild mushrooms for their promising potentials. Okay, so this CLSU project or this CLSU Tuklas Lunas program is composed of 4 major projects. So the first project focuses on the collection, entomological study, as well as on the optimization of the culture conditions of the Philippine mushrooms in order to produce the massive biomass, mass produce the biomass of the mushroom, which is intended for the, of course, the screening of their biological activities, among other attributes. So the project 2 focuses on the extraction of the bioactive compounds, okay, and then subject these to screening using the enzyme-based assay, particularly on the anti-diabetic, anti-inflammatory, and anti-hypertensive activity of the mushroom. So for the project 3, this is focused on the this focuses on the molecular identification as well as on the toxicity profiling and the anti-cancer activity of the extracts of Philippine mushrooms. Whereas the project 4 is focused on the development of mushroom-based food products as well as on the market study and the pre-commercialization of the developed mushroom-based food products. So in our mycological expedition of the selected areas in Luzon, Philippines, so this is one of the major islands of the Philippines in Luzon, so a total of 585 wild mushrooms were successfully collected from the wild and bring them into the laboratory for their, optimization study to rescue their mycelia for the other applications or for the screening activities. Okay, so these were collected in the different regions of Luzon Island. So we have the Central Luzon, Ilocos region, Cagayan Valley, the Calabarzon, and the Bicol region. And then as prelude to their effective utilization, so these mushrooms were, of course, obtained their germplasm. Okay, so in laboratory, we have this cryogenic bank where we actually preserve our cell lines in order for us to, the rescued mycelia will not be, will of course rescue the mycelia and preserve the cell lines. And then, of course, we also established the germplasm, the nucleotide sequence-based identity prior to its, of course, identification, prior to its elucidation of the bioactivity as well as on the screening process. So with our continuous effort on the collection of wild mushrooms, so we have successfully the only germplasm collection of Philippine wild mushrooms in the country. So we are the only repository of the mushroom germplasm in the Philippines, and this is located at the Center for Tropical Mushroom Research and Development. And out of 585 collections of mushrooms, we have a total of 388 molecularly identified mushrooms using the ITS sequence-based primer. And the results of our molecular identification, as well as the species listing of these mushrooms, Philippine wild mushrooms, were published in Scopus Index Journal, including the Biocatalysis and Agricultural Biotechnology, the Journal of ISAAS, and the International Journal of Pharmaceutical and Allied Sciences, among other journals. Of course, after confirming the identity of the Philippine wild mushrooms, so we do the optimization of the culture condition for both mycelial growth as well as for both mycelial growth in the fruiting body in order for us to mass produce or to, of course, obtain the mass production of the mycelia as well as the fruiting body for the extraction and the screening process. And we were successful to establish the production technology of Ganoderma lucidum. So as you can see in this picture, we have the wild and the domesticated one, meaning when you say domesticated, this is actually growing under laboratory condition. And then we have the Pleurotus sidiosus, so we have the Lentinus tigrinus, we have Pleurotus jamor, our pink lady mushroom. So this was collected in one of the province in, I think, in Maria Aurora province in Luzon Island, Philippines. So, of course, the most common mushroom in the Philippines, the Bulbariella bulbacea. We have also this giant mushroom. We call this as Colibia rinaciana, its scientific name, but commonly this is kabuting kalaw. Okay, so for among Filipinos. And we have also Coprinopsis cinerea. So this is a weed fungus. Okay, so when you say weed fungus, this is a contaminant, okay, of the Bulbariella bulbacea. However, we did some investigation on the development of its practical innovative technology using a small substrate in a bottle. Okay, so we have also the Schizophyllum commune, Lentinus saharcaju, Lentinus strigosus, Odimentiella canarii, and Pagnolus antillarium, and of course, many other species of mushrooms. So all in all, a total of 143 mushrooms were successfully optimized in our laboratory, and 105 mushrooms successfully produced the fruiting bodies using the rice based substrate formulation, and 28 mushrooms were, of course, mass-produced the mycelia using a submerged fermentation process. Okay, so results of the optimization study were published also in Scopus Index journals. Okay, so we also applied 6 utility model for the newly recorded mushrooms in the Philippines. We have applied 6 utility model relating to the composition of the substrate as well as on the process of the cultivation of this mushroom. Okay, so we have 6 utility model for this, and then we also develop and publish books. Okay, so this the first book is the Pictorial Guide on Philippine Indigenous Mushroom, and the second book is the Ethnomycology and Microfarming of Philippine Mushrooms, which serves as a reference material for mushroom producers and mushroom farmers in the Philippines. Okay, so after the cultivation process, okay, so as I did mention a while ago, the biomass or the mushroom biomass will process this will be extracted, or this will be extracted for the bioactivity elucidation, and for the bioactivity we are screening the activity for its anti-diabetic activity, anti-hypertensive, anti-inflammatory, as well as the anti-cancer activity of the different species of mushroom. And we have successfully demonstrated the biological activity of the different species of mushrooms. So we have produced a total of 69 ethanol extract and 46 hot water extract. And amongst of 69 ethanol extract, we have 61 species of mushroom exhibited anti-diabetic activity, 10 anti-inflammatory activity, 2 anti-hypertensive, and 7 cytotoxic activity. On the other hand, for the hot water, out of 46 species or extract, we have 45 for anti-diabetic. We have 1 anti-hypertensive and 4 cytotoxic or anti-cancer, showing anti-cancer activity. Okay, so I was also given a chance to pursue my fellowship or Fulbright scholarship program at the USDA. Okay, so U.S. Department of Agriculture, to do with the submerged fermentation of the Philippine wild mushroom. So we have successfully demonstrated here the mycelial biomass production of the Lentinus, the Aganoderma lucidum, the Bulbariella bulbacea, the Pleurotus species, and the Schizophyllum commune. And this is actually for the lipid, what do you call this? Functional lipid production. Okay, so functional lipid production, so meaning they have we extracted the lipid composition of the mushroom, okay, and then subject to the different bioactivity evaluation. And we found out that fraction 2 of the Schizophyllum commune showed activity against Staphylococcus aureus. Okay, so this is actually this is also a published paper. Okay, so we also characterized the different lipids from mushrooms, and we also published the first report on the identification of molecular species of the acyl glycerols of the Philippine wild mushrooms, particularly on the Ganoderma lucidum. So in this paper, we reported 40 molecular species of acyl glycerols which contain fatty acids with odd-numbered carbon atoms. Okay, and then of course we are also lucky to have a collaborator in UTMB, University of Texas, and the Anderson Cancer Center in Houston, Texas, to investigate the cytotoxic activity of those Philippine wild mushrooms that we collected within the vicinity of the Central Luzon State University. And we found out that among these mushrooms, four samples or four mushrooms exhibited very low IC50 value, meaning they show the strongest activity against cancer cell lines, okay, or hematologic malignant cell lines, okay. So in this case, we have tested in the AML, or the acute myelogenous leukemia, and we found out that the red, okay, so this red, which I coded red, this is actually your Gymnophilus purpureus formulosus. Okay, this is a Philippine wild mushroom, and this displayed the strongest activity against the AML cell lines, followed by the OCA, or Artemisia canarii, and the two strains of Ganoderma lucidum. Okay, so consistent with the three lymphoma cells, consistent that red showed or displayed the lowest activity, followed by OCA and the two strains of Ganoderma lucidum. Okay, so again, in terms of the multiple myeloma cell lines, again, Gymnophilus purpureus formulosus, or the red, showed the lowest IC50 value. So given their notable cytotoxic activity, so this is the Gymnophilus purpureus formulosus, this is the red, and we have the white one at the center, the center picture, and this is the Artemisia canarii, and then the picture in the right is actually the Ganoderma lucidum, CLSU, and these mushrooms were subjected for mechanistic investigation. Okay, and for the mechanism of the cytotoxic activity, our findings are actually published in International Journal of Medicinal Mushroom. Okay, so we also reported the first ever activity of cytotoxic activity of the Gymnophilus purpureus formulosus, and we published this in both hematologic malignant cell lines and patient-derived cell lines, and some underlying mechanism of cytotoxicity were highlighted in this paper. Okay, so we proposed the model for this observed cytotoxic activity of Gymnophilus purpureus, focusing on the activation of the ASK, MEK, SAPK, JNK pathway as one of the major mechanism of the cytotoxic activity. We also analyzed the nutritional evaluation. So say, for example, in this picture, so you have, in this table, you can see the different stages of the Bulbariella bulbacea, showing different nutritional attributes. Okay, so meaning different stages of Bulbariella show different nutritional attributes. Okay, so we also established the standard amino acid profile of the 5 species of mushroom, and then non-standard amino acid activity. And then in that case, these mushrooms that showed biological activities as well as nutritional attributes, these were the priority for the product development of mushroom-based and functional food products. And based on our prioritization study, we listed the five priority mushrooms, which are including the Lentinus tigrinus, Lentinus spirosulus, Ganoderma lucidum A, B, and Pleurotus cysticiosus. So we also look at the nutritional as well as the elemental composition of this mushroom. So the first product in line is the mushroom snack chips. Okay, so our mushroom chips. These are the nutritional composition as well as the amino acid profile. We have the shinoline. Okay, this is common to Filipinos, but we call this mushroom stick snack. Okay, so these are the nutritional as well as the amino acid profile. We have torikake, or the mushroom-based seasoning flakes, and this is actually considered as rice seasoning. Okay, so there we have the nutritional as well as the amino acid profile of this product. And then we have the mushroom instant tea. Okay, so mushroom instant tea. Okay, so that's actually a powder or extract of the mushroom. And then since Ganoderma lucidum is not palatable, we tried to develop a ready-to-drink mushroom-infused soy coffee. Okay, so similarly, we have the nutritional composition as well as the amino acid profile. So these five mushroom-based products are considered microbial stable, and it has high consumer purchase intention, and of course, it contains essential amino acid. Okay, now let's move to the functional food. So when you say functional food, aside from the nutritional attributes, it has nutraceutical attributes. Okay, so say for example, we have the mushroom composite seasoning base and the spray dried mushroom powder. With this spray dried mushroom powder, we have this actually last 2 months, 2 weeks ago, we had the meeting with the University of San Agustin in order for us to encapsulate this spray dried mushroom powder of these different species of mushroom. Okay, so we consider this functional because we found that this product is active against alpha glucosidase. It contains essential amino acid, and of course, it have high consumer purchase intention. We also tried to, of course, the team also provide the posters for the introductory advertising campaigns of this product as part of the soft launching. Okay, so these developed mushroom-based food products were already applied for, of course, utility model applications. So including the product itself as well as the process of developing the product. We filed the as a utility model application. Okay, so looking forward, so with this mushrooms, we have to, of course, broaden the mycological expedition. So we propose to NRCP, National Research Council of the Philippines, and not just to look at the Luzon Island, but to, of course, to consider also the Visayas and Mindanao region. And of course, we want to isolate and characterize the bioactive compounds responsible for the demonstrated biological activity and commercialization of the product through our, of course, with the help of our university. With that, I would like to thank the Philippine Council for Health Research and Development, or PCHRD, of the Department of Science and Technology in the Philippines for providing us our research fund, and of course the UT University of Texas and the Anderson Cancer Center in Houston, Texas for the collaboration, research collaboration. Thank you very much.
Okay, thank you, Professor Dr. Rich Milton Dule, for the excellent presentation and sharing about mushrooms. Let's now have a short Q&A sessions from the audience.
Hi, I'm Celine, one of the students from UMPSA, and I would like to ask what are the ways you recommend to prepare or to cook mushroom at home in order to preserve the nutrient and bioactive compound in mushroom?
Yeah, actually, if you want to really if you want to really get all the nutrients, the vitamins, do not cook. You eat fresh. However, we usually add the mushroom to vegetables, meat, and among other, as a mix to as a mix or flavor enhancer for the different viand or for the different food in our country. Okay, so for of course, in regarding the process, drying is the most best way to preserve the nutritional composition as well as the, of course, other attributes or properties, biological properties of mushroom. Thank you.
Okay, thank you, Ms. Celine, and thank you again, Professor Dr. Rich Milton Dule, for your sharing and answering the question.
Thank
Okay,
you.
now we have settled with 6 remarkable sharing from different expertise regarding sustainable biotechnology for food security. Let us now hear from Dr. Nazira Mahmood, Head of Industrial Biotechnology Program, this UMPSA on Introduction to Program Industrial Biotechnology. Please welcome.
Thank you very much, Dr. Aisha. Hello, everyone from UMPSA and from CLFU. I will share my slide first. All right, so I am Nazira, currently the head of program for Industrial Biotechnology. Under the program, we are currently offering Bachelor of Applied Sciences in Industrial Biotechnology with honors. So I would like to start with a bit introduction about UMPSA. University Malaysia Pahang Al Sultan Abdullah, UMPSA, established as one of the technical university in Malaysia in 2002. We are offering variety of engineering and technology-based programs, including high-level TVET. We are currently the best university in the Malaysian Technical University Network, MTUN. So this is our university top management. We are led by our Vice Chancellor, Professor Dr. Yatimah binti Alias. So the vision of the university is to be a distinguished technological university, and we are determined to excel in providing community-driven education, research, and high-level technology services through a social entrepreneurship ecosystem. So there are two campuses of UMPSA, being Pekan campus is the main campus. So Faculty of Industrial Sciences and Technology is located in another campus, which is Gampang campus. So moving on to the Faculty of Industrial Sciences and Technology, this is the faculty management. We are led by our Dean, Associate Professor Dr. Aizi Nur Mazila binti Ramli. Helping her several deputy deans and head of programs. So Bachelor of Applied Sciences Industrial Biotechnology with honors was first offered in 2009, during which at that time the approval as well as the full accreditation was under Malaysia Qualification Agency. In 2022, we are moving to the professional body for our accreditation, Malaysia Board of Technologies or MBOT. So this is a 4-year program, including 6-month industrial training that students have to take at the very end semester of their study, which is semester 8. So we are accepting students from various backgrounds. Local students need to have STPM, either STPM matriculation or foundation certificate, including a diploma from private or public university. So we are also accepting international students provided that they have high school certificate, or O-level or any equivalent certificate, including diploma and equivalent. So we have provided a good number of skilled graduates in the field of biotechnology ever since the first graduation in 2013. So we have more than, if I'm not mistaken, 2,000 graduates that have been working locally and globally in various biotechnology-based companies. So this also includes food, agriculture, manufacturing, and also pharmaceutical-based company. So I would like to share just a bit on our curriculum. Students in Bachelor of Applied Sciences in Industrial Biotechnology with honors are taking all of these courses that are divided into program core, faculty core, and university core. With overall practical component of around 54% and theoretical component of around 46%. So towards the end of their year, around semester 6 or 7, students need to take elective courses that will help them to kind of tailor their study or directed their knowledge in certain field of biotechnology. These are the list of elective courses that are currently being offered in the faculty. So teaching are conducted in various ways. So we adopt various teaching style to help in classroom learning. We also have online learning that often conducted synchronous or asynchronous via our online platform. We also have practical or lab classes. We also have several in-class teaching initiatives that we call the first one co-lecturing, where we invite industry experts to classroom to give lecture to students with the aim to expose students on the real application of certain knowledge. We have global classroom, where we invite a lecturer from overseas to give lecture to students through online platform with the aim to give exposure to students on maybe different teaching style or situation that specific field that they are learning that happen in the other country, for example. And students need to take final year project at the semester 6 and 8. And we also have out of the classroom initiative, we call that industrial visit, where we bring students to industry, student visit to certain industry with the aim to let the students see the real thing that happening in industry. So these are some of the out of the classroom activities that happened over the past semester. Students have went to various industry for industrial visit. So we also encourage students to participate in any competition out there. We also have an initiative that we call that SULAM. Okay, SULAM is Service Learning Malaysia University for Society, that where we integrate student and community as part of the teaching and learning process. So these are some of the picture of co-lecturing with industry experts that have been conducted over the past semester. So the faculty members of Industrial Biotechnology Program also have a lot of collaboration with international universities. Through that collaboration, we often do a global classroom. We even organize summer school, a colloquium. We invite our partner to co-organize a conference. So for your information, Industrial Biotechnology Program under our research cluster Biosphere organizing one conference, this conference IEC once in every 2 years. So these are some more picture of the activities with our international collaborators. If you can see, these are some of the international university that we have collaboration with right now. So we also encourage our students to go overseas, okay, for student exchange or mobility programs, either self-sponsored or through monetary funding with university, from the university, and vice versa. We also hosting multiple mobility students from overseas. So they are coming from countries like China, Thailand, Indonesia, and Kazakhstan. So one of the difference, or I would say one special point of Industrial Biotechnology Program in UMPSA, we are also offering this dual degree program with Kazakh's National Agrarian Research University, Kaznaru in Kazakhstan, where students have a chance to enroll in this program, and students that enroll in this program, if they pass all of the assessment that are given by both of the university, eventually they will be awarded by 2 degree scrolls, which are one from UMPSA, Bachelor of Applied Science in Industrial Biotechnology with honors, and one from Kaznaru, Bachelor of Techniques and Technology in Biotechnology. So research activities that happen in the Faculty of Industrial Sciences and Technology divided into these 4 different research clusters. We have Biosphere, those research that are related to biological sciences. We have AFM, Advanced Functional Materials, Chem-AT, Chemistry for Advanced Technology. We have Occupational Safety, Health and Environment, OSHI. So most of the research of Industrial Biotechnology members is fall under Biosphere Research Cluster, with some of the research area as listed over here. Okay, you can see Dr. Aizi is a picture of Dr. Aizi and Dr. Nina is there. So moving forward, so we are looking forward for these activities that we have today between UMPSA and CLFU to kind of have more meaningful, more impact in the future. So we are looking forward for collaboration in terms of academic, for initiatives that we call as global classroom, as I mentioned before, or even mobility or student exchange program. So we also looking forward for collaboration in terms of research through research grant application. For example, one specific grant that we have as known as International Matching Grant, where we have same contribution from the both institution to achieve specific set of outcome that is agreed on. So of course, in terms of publication and maybe in the future, we could co-organize a conference. All right. So with that, that's the end of my presentation. Thank you very much for your attention.
Very interesting program advertised by Dr. Nazira about Industrial Biotechnology, UMPSA. And now I'm open to the floor if there is any question regarding industrial biotech. Okay, so if there is no question, I will proceed with the last but not least, our eighth presenter and last speaker. So thank you, Dr. Nazira, for the presentations. Now I'm calling upon Associate Professor Dr. Hazel Monica Peralta, Acting Dean, College of Fisheries, CLSU, for introduction to academic activities of Central Luzon State University. Please welcome.
Okay. Good morning, everyone. May I request the technical team to please show the short video which I shared in the Google Drive, the CLSU at a glance, please.
Yes, Secretariat will come up to it shortly.
Yeah. So after the video, I will also have a short explanation for what the academic affairs is offering for everyone.
Okay, so for technical team, is it okay to share the video?
Kejap, kejap eh.
Central Luzon State University, through the years, has grown into a comprehensive and inclusive higher education institution that transforms the mind and improves the quality of life of peoples and communities it serves. From its humble beginning as an agricultural school, with its vast land resources, CLSU offers a conducive environment for learning, research and production catering the needs of students, faculty, staff, researchers and other stakeholders. For more than 100 years, agriculture has remained the flagship program of CLSU, keeping a multidisciplinary approach through the complementation of the different curricular programs it offers, including arts, social sciences, business management, and other allied fields. CLSU is also recognized as an agri-tourism site in Luzon, featuring the different R&D centers that specializes in precision agriculture, livestock, crops and fisheries. These research centers support, strengthen and sustain the thrusts of the university's academic program. CLSU being strategically located at the heart of the Muñoz science community, it has access to national R&D institutions bringing in additional resources, facilities and experts, which further amplifies the research initiatives of the university. CLSU today stands proud as one of the renowned and prestigious universities in the Philippines and Southeast Asia, and is positioning itself in the global community. CLSU, keeping agriculture as a way of life.
Okay, thank you very much. There's another presentation. It's a PowerPoint presentation, the academic affairs. Can you help me share it? Okay, thank you very much. So just very quick, next slide, please. Okay, so this is the Central Luzon State University overview. Okay, next slide, please. Okay, so the Central Luzon State University is located at the Central Luzon. No? So we have 3 islands in the Philippines, big islands, the Luzon, Visayas, and Mindanao. So the Science City of Muñoz is in the Central Luzon. And we have a population of approximately 13,500 or near 14,000, both undergraduate and graduate students each semester. And we have around 550 academic staff, faculty members that are committed to quality education and research. So not only teaching, but we also have research. Next slide, please. Okay, so our university is headed by our university president, Professor Dr. Evaristo A. Abella, and we have 4 executives under him. The next slide, please. We have the Vice President for Academic Affairs who give the talk earlier in the morning. Okay, and we also have the Vice President for Administration, Vice President for, next please, Vice President for Research and Extension, and another one is the Vice President for Business Affairs. Next slide, please. We have 10 colleges. This is equivalent to your faculty. No? So here we call it college. So we have College of Agriculture, College of Fisheries, College of Arts and Social Sciences, College of Home Science and Industry, College of Business and Accountancy, College of Science, College of Education, College of Veterinary Science and Medicine, College of Engineering, and we also have the Distance Open and Transnational Education University. This is it's like an open university, so most of the classes are done online. Next, please. Okay, so we have an offering of 32 undergraduate programs or the bachelor's program, and we have 23 master's degree programs and a total of 13 doctoral degree programs. Next. Okay, so this is the College of Agriculture. So when you enter, this is what you will see. No? So next slide, please. So there are 6 departments in the College of Agriculture offering both undergraduate and graduate programs. So we have Department of Soil Sciences, Department of Agriculture Extension Education, Department of Animal Science, Department of Agri-Management, Department of Crop Protection, and Department of Crop Science. Next slide, please. We also have the College of Arts and Social Sciences with 5 departments offering also both undergraduate and graduate programs. Department of Filipino, Department of English and Humanities, Social Science, Communication and Development Studies, and Department of Psychology. And next is the Business and Accountancy. So business and accountancy offer different programs as well, but there are only 2 departments, the accountancy and business. Next is the College of Education that offers also both graduate and undergraduate programs with 5 departments, namely Department of Early Childhood and Elementary Education, Department of Education Policy and Practice, Department of Language, Culture and Art Education, Department of Science Education, and Department of Technology, Livelihood and Life Skills Education. Next is the College of Engineering, which offers also both undergraduate and graduate program with 4 different departments: the Department of Agricultural and Biosystem Engineering, Department of Information Technology, Civil Engineering, and Department of Engineering Science. Okay, next is the College of Fisheries, where I belong. There are 3 departments offering Bachelor of Science in Fisheries and Masters and PhD in Aquaculture. We have the Department of Aquaculture, Aquatic Post-Harvest, and Aquatic Resources, Ecology and Management. Next, we also have the College of Home Science and Industry offering different programs with the departments. Next slide, please. The Department of Textile and Garment Technology, Department of Hospitality and Tourism, and Department of Food Science and Technology, where our guest speaker, Dr. John Juvenal, is teaching. Okay, so next, please. Okay, so we also have the College of Veterinary Science and Medicine, offering 3 departments: the Department of Basic Veterinary Sciences, Department of Veterinary Paraclinic Sciences, and Department of Veterinary Clinic Sciences. And lastly, next. Okay, this is last for the offering of face-to-face module. Okay, so the College of Science with also, next slide please. There are 5 departments: the Department of Statistics, Department of Chemistry, Department of Biological Sciences, Department of Mathematics and Physics, and Department of Environmental Science and Meteorology. Okay, and last but not the least is our it's like Open University. We call it CLSU Distance Open and Transnational University, offering different programs such as, next slide please. Okay, so next slide please. So we have the Masters of Science in Education, major in Education, Educational Management, major in Mathematics, and Development Education. So next please. We also offer in our .uni the Rural Development, Doctor of Philosophy in Rural Development, Masters in Agribusiness Management, and Masters of Business Administration. Next slide, please. Okay, and also the .uni is offering Masters of Science in Renewable Energy Systems, Doctor of Philosophy in Sustainable Food Systems by Research, and also the Master in Environmental Management. So for further information, next slide please. Okay, you can visit us in our website, clsu.edu.ph. And if you want more information, you can email info@clsu.edu.ph. Okay, so thank you very much.
Okay, super briefing from CLSU. And now I'm open if there is any relatable question to CLSU. If there is any. Okay, so thank you again to Associate Professor Dr. Hazel Monica for sharing session about CLSU.
Thank you.
As we come to the end of this enriching session, I would like to call upon the Director of COA 2025 from UMPSA, Associate Professor Dr. Tan Swat Hian, to give closing remarks. The session is yours. Sorry, Dr. Tan, we cannot hear you. Oh, okay.
Okay. Salam sejahtera and very good afternoon. Yang Berusaha Associate Professor Dr. Aizi Normazila binti Ramli, Dean of Faculty Industrial Science and Technology. TS Dr. Azizan bin Ramli, Deputy Dean, Research and Postgraduate Study. TS Dr. Hanida binti Abdul Aziz, Deputy Dean Academic and International Affairs. TS Dr. Muhammad Hafiz bin Maswi, Deputy Dean Student and Alumni Affairs. Our esteemed colleagues from Central Luzon State University, Professor Dr. Avelina Velasco, Vice President of Academic Affairs, CLSU, Associate Professor Dr. Hazel Monica Meshes, Prata, Acting Dean, College of Fisheries, distinguished speakers, respected participants, faculty members, students, and guests. On behalf of the organizing committees, it is my great honor to deliver the closing speech for the 5th Colloquium of Academic and Research COA 2025. Today marks the successful conclusions of an event that has not only showcased the depth of research and expertise within the field of biotechnology and agriculture, but also furthered our shared visions of creating impactful academic collaborations across borders. Allow me to express these sentiments through a simple verse. The rose blooms with fragrance grace, bright and fresh in the morning's face. These colloquiums end with a meaningful trace, knowledge share, progress and praise. This year theme, Sustainable Biotechnology for Food Securities, has brought together brilliant minds from UMPSA and CLSU, as well as speakers and participants who have enriched the discourse with their insight, perspective, and innovative solutions. Over the course of this colloquium, we have achieved several key milestones. Knowledge exchange. Through presentations and discussions, we have explored critical topics in food science and technology, bioprocess and enzyme technology, and natural product and biomedical technologies that are essentials for global food security and sustainability. Strengthens partnerships. This inaugural collaboration with CLSU has set a strong foundation for future academics and research initiatives, including student exchange program, joint research projects, and global classroom activities. For community buildings, the event has fostered invaluable networks among researchers, academics, and students from diverse institutions, paving the way for continued dialogues and shared learnings. I would like to extend my deepest gratitude to our distinguished speakers whose expertise and insights has been pivotal to the success of these colloquiums. Your contributions have inspired us to think critically and innovate boldly. To the participants, your active engagement, thoughtful questions, and collaborative spirits have made this event truly dynamic and impactful. We hope you will carry the knowledge and connections gained here into your future endeavors. To our co-organizers, faculties members, and committee teams from both UMPSA and CLSU, I commend your dedications and hard work in ensuring the seamless executions of CORE 2025. Your commitment reflects the true essence of academics and professionals' excellence. As we conclude this event, let's reaffirm our collective commitment to advancing research and educations that address global challenges. Let us also recognize the potentials of this partnership to achieve even greater accomplishment in the years to come. With that, I officially declare the fifth Colloquium of Academic and Research CORE 2025 closed. Thank you, and I wish you continued success in your academics and professional journeys.
Okay, thank you to Associate Professor Dr. Tan Suat Hian. With the end of the speech, I kindly ask all speakers and participants to turn on your cameras for our second group photo informal session. Our technical team will take a screenshot for a while.
I think most of students did not switch on their camera. Please switch on the camera, please. Okay, satu, dua, tiga. Another time. One, two, three. One more time. One, two, three. Smiley face. Okay, thank you.
Okay, a heartfelt thank you to all the speakers and members who joined us through Google Meets, YouTube Live, and Facebook Live today. We kindly remind all attendees to complete the registration form and evaluation form for UMPSA staff, students, and also all the participants before the end of today's session. Thank you for all your cooperation, and we hope that COA 2025 has sparked new ideas and collaborations aligned with our team's sustainable biotechnology for food security. On behalf of the Faculty of Industrial Sciences and Technology UMPSA, we look forward to continued academic and research cooperation with CLSU. Until we meet again in future initiatives, thank you and have a wonderful day. Stay safe and goodbye. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.